My name is Michael Goldberg. I'm the executive director of our Veal Institute for Entrepreneurship here at Case Western Reserve University. I'm also a professor teaching entrepreneurship at Weatherhead. It is great to welcome so many great students here in person. You know, we are back in person at the university. And also, not surprisingly, because the way life is these days, we're doing things on Zoom. So thank you for those on Zoom for your patience. It took a few minutes to get things going, but without further ado, I want to introduce Jessica Scarborough, who's going to come up and introduce Will to kick things off. Jessica is one of our awesome students who just finished the translational fellowship program. She'll tell you a bit about herself and then introduce Will. And I know, in knowing Will, and it's funny, I'm wearing almost like the Will, for those of you like saw his picture, he's like in a vest. He's usually, you know, outdoors. He wants this to be super interactive. So I'm encouraging those who are here in person and on Zoom, please let us know if you've got questions. And I want to thank also our colleagues from Dartmouth who are joining. Alexis Abramson, who's our former colleague here at Case Western Reserve and is now your colleague as the Dean of Engineering at Dartmouth, gave a shout out and said how awesome you guys are. So we're so thrilled to have you here. But I will shut up and turn it over to Jessica. Thank you, Jessica.
Hi, everyone. As was just stated, I'm Jessica Scarborough. I'm an MD, PhD candidate here at Case, and I also am a Translational Fellows alumni. And during my time as a fellow, I worked on a cisplatin sensitivity signature for use with bladder cancer patients for figuring out who's going to respond best to their chemotherapy treatments. And I'd like to start by welcoming our new students from the School of Medicine Health Innovation Pathway Program led by Joe Jankowski. Awesome to have you guys here. Welcome. And thank you to everyone here for coming to the first in our three-part series of the talks led by Will Lewis, who I'm really excited to introduce you today. Will is a native Clevelander who graduated from CWRU with a Master's of Business Administration and Juris Doctor with honors in 1996. And he has over 20 years of executive experience and a 20-year history of success in the pharmaceutical and finance industries. Most recently, Will joined Insmed in 2012 as the president, CEO, and a member of the board of directors. And in 2018, he became the chair of that board of directors. Insmed is powered by purpose as a biopharmaceutical company which develops and brings to market therapies that help treat patients with rare and serious conditions. Will also currently serves as the chair of the Board of Trustees of BioNJ, which is the Life Sciences Association of New Jersey, and he's also a member of the Board of Trustees of CWRU and the Board of Trustees of the Helix Acquisition Corp. So please join me in giving a warm welcome to Will Lewis.
Thank you very much. Your mic.
The mic. Hopefully this will pick up.
Yeah.
The first thing I want to do is say thank you for your work with cisplatin, because my father had bladder cancer before he passed. And folks, that is what this is all about. It is about finding therapies and ideas and bringing them forward to change the outcome of people's lives who are either born with or inherit a raw deal at some point. There is nothing, I can tell you, more gratifying than being a part of that system that creates ideas and turns them into therapy. That's what biotech is all about. It's what pharmaceuticals are all about. And don't let anyone tell you otherwise. I keep a stack of letters on my desk. I have at my last company and at this one. They're a short stack from patients that are describing the positive outcomes they've experienced as a byproduct of the work that we've done. Now, there is no more inappropriate recipient than me for these letters, but they send it to me because I happen to be the person that is the spokesperson for the company. But the real work starts with the idea that's generated by folks like you. And today what we're going to do is talk about how we take those ideas, how we identify what might have impact, and how we turn that into something that can have an impact on a patient somewhere. I will tell you that this is going to be a productive day because I have two colleagues joining me, Chris and Carl, who are not on the screen, but they're out there. Maybe you can see them on the ether. And in a minute, I'm going to ask them to introduce themselves. But let me just tell you a little bit about Insmed so you have some idea of how quickly things when they hit can go. And I want to try and frame out some of the challenges that we face as we think about tech transfer and idea generation. So when I joined Insmed 9 years ago, there were about as many employees as there are people in the room. So 30 or fewer. Today it's 600 employees. We have offices in US, Europe, and Japan. This is 9 years later, on the heels of one drug that got approved, the first of its kind to treat a rare condition called refractory non-tuberculous mycobacteria lung infection. Prior to the arrival of this drug, there was nothing approved to treat it. It's actually a drug-device combination, and we are super proud of this impact. It's actually the second company I've started. The last one was myself and 2 other people, and we ended up getting that drug approved to treat something called homozygous familial hypercholesterolemia. What I've found is most important when thinking about the ideation around biotech and what you can do is identifying the clear unmet need that you hope to address. And as big an idea as that may be, whatever your area of expertise, whatever your area of interest, you've got to start off with the problem you're trying to solve. And in our quest to find those ideas in industry, we're constantly talking to folks like you who have ideas. Without that bridge built, we go nowhere. Some of this stuff we can generate internally by hiring people and getting the company to do its own work. But most of the time, the most interesting ideas come from academia or academia that transfers them into the early part of the research in industry. My first company was started when I in-licensed a program from the University of Pennsylvania. This is a program that had been donated by a big pharma company. They didn't think it was going to go anywhere. They didn't think it was worth anything. And so they gave it to the University of Pennsylvania. And so we in-licensed it from them for almost nothing. And then we turned that into a public company that ended up employing hundreds of people. And most importantly, ended up giving folks that had this HoFH condition some hope. So in our current state, Insmed is now, with its approved product, built a pipeline behind it. But we are really at a point where we want to break out and we want to do something really disruptive. And now we have the luxury of trying to do that. It's one of the most exciting things about being part of a company that has success is that it gives you the tools and the opportunity to go and have really disruptive impact that maybe you might not otherwise be able to go after because it's too risky or it's too early. So today we're joined by Chris and Carl, and I'd like them to introduce themselves. What has happened at Insmed is through serendipity, we managed to, the smarter people at the company than me, managed to run into the work that Chris and Carl were doing. And we started a dialogue with them. They were professors at Dartmouth and both have fascinating backgrounds and areas of study, research that has gone on for many, many years. And we decided to pool our resources collectively and work together to try and solve some of these problems. So Carl or Chris, whichever one of you wants to go first, please introduce yourselves.
I guess I got the nod. Hi, everyone. Nice to join you virtually. My name is Chris Bailey Kellogg. I'm now the Executive Director of Computational Biology at Insmed. As Will mentioned, I was a professor at Dartmouth in the Computer Science Department. My background and much of my academic career is in computer science, transitioning to computational biology. I don't know how much intro you want now, Will. Is that enough, or do you want me to go a little bit further?
Work that you do so that people understand some of the research that you've done and how that ended up linking to what we're doing now. Just a couple of minutes, maybe.
Okay, a couple of minutes, yeah. So as a computational biologist, I was always interested in trying to understand at the molecular level, how do proteins work? How can we program those proteins to do what we want them to do? So that involves modeling the proteins, that involves optimizing properties of the proteins and so forth. And so when Carl joined Dartmouth, we struck a collaboration that kind of is the bedrock of the whole rest of the story we'll be talking about today, and looking how computational methods can be used collaboratively with protein engineering techniques to redesign therapeutic proteins for improved properties. And so my academic career has really been focused on using those computational techniques, developing new computational techniques, and then in collaboration with Carl and others, putting them into practice. That led us to start a small company, Stealth Biologics, that we'll probably talk about a little bit more today as well, where we started moving some of the ideas that we proved the validity of academically, started moving them and applying them into actual practice in collaboration with a number of different biotech and biopharma companies. And that started snowballing, eventually leading us to Insmed, which, again, I don't want to tell the whole punchline now, so I'll stop with the intro and let Carl chime in.
Yeah, so good afternoon, everyone. I'm Carl Grissold. I'm the executive director of
Hey, Carl, can you hold on for one quick second? Okay, you guys are back up.
We lost you for a minute.
Okay. You guys have me now?
We do. Thanks.
Okay, great. So, I was just saying good afternoon, everyone. So I'm Carl Grissold. I'm the executive director of biologics research at Insmed, which is a thrill for me to have stepped into this position. I've been a professor at the Thayer School of Engineering at Dartmouth, and that was kind of the genesis of the collaborative work that Chris and I did together in this area of protein engineering. So I've been interested kind of during the majority of my professional and academic career in the applicability or practical utility of proteins, right? These just phenomenal nanomachines that are the basis for life. How can we actually leverage those proteins to solve practical real-world problems? And in particular, I've just become fascinated with the idea that we no longer have to rely on the repertoire of proteins that are present kind of in the natural biosphere, in the global proteome, but instead we can actually create new versions of those proteins, or potentially even proteins de novo that have kind of finely tuned properties and high performance characteristics in practical applications. And I think a longstanding interest of Chris's and mine has been specifically the application of proteins in treating disease. So again, that's the basis of a lot of our collaborative work. And my role in this effort is to help figure out how to leverage some of the computational design and the guidance that Chris's computational team provides, how to leverage that in the laboratory and how to render these molecules and bring them to life and validate those molecules and find the best candidates that come out of the design process and hopefully candidates that have the potential for, again, translation and getting into the clinic and hopefully making a difference in patients' lives.
Thanks, both Chris and Carl. So one of the things I want to do before we go into maybe a further discussion of the issues and challenges that they faced is talk about a common language in terms of what we're up against here. So you may have an idea that relates as theirs does to the ultimate impact your idea may have on disease. But what is the journey that must take? The first thing you have to do is come up with the idea, right? And you think, okay, well, that's the real challenge. And certainly that's what history would teach us is where the most difficult hurdle lies. But it's just the beginning of a very difficult and long-term process. And I don't want to dissuade anyone by going through this, but it's really important that you understand how enormous a hurdle it is to take a really good idea and take it all the way through to the place where it will have impact on a patient. And the reason I started out with that compass point of thinking of the patient is because that is the thing that is going to pull you through when you are struggling along the way. And you will struggle, whether you hand it off to someone else and ask them what progress they're making, or whether you choose to try to bring it forward yourself. There is an immense challenge in front of you. The good news is, once the idea is identified and the ecosystem of the for-profit industry world pays attention, you will be swept up in that experience. And while you will lose some sense of grounding, disorienting, the fact is enormous resources are brought to bear in support of what you are doing. And that's what you're after. You're trying to get this to a place where the people on the outside who are skilled enough can identify and say, this is something we need to turn our attention and our capital and our time to. So what does that look like? Well, if we're talking about a therapeutic, one of the first things you have to do is, once you have the idea, you got to protect it. Intellectual property. So it's one of the most important things that you can do to ensure that all the work that's going to come will actually be something that that ecosystem will pay attention to. Because if it is something that's novel and innovative and it's published where everyone can read it, unfortunately, at that moment, it absolutely deserves attention, and the system in which we all work will not give it to it, because it will not have a way of getting back what it's going to have to put in to bring it all the way to fruition. What does that look like? Once you have your intellectual property filed and someone decides they want to support that, you're looking for early capital, right? We're talking hundreds of thousands to millions of dollars in the earliest stages just to get through the place where you have gotten a drug ready to be filed with the FDA, so-called IND. That IND filing is the kickoff of the clinical work you will need to do. To get to an IND, you have to run animal model studies. You have to do ex vivo study work to make sure that what you have is safe to put in a person. And the FDA will keep a weather eye on that very carefully. So here's the thing that is counterintuitive about biotech. The creativity is in this room and whoever is dialed in. Once that drug, once that idea gets on the track to go through to approval, you can't change it, because all of the testing you're doing is to validate and provide assurance that what you have done is safe and efficacious. The moment you change it, you got to go back to the beginning. And what does the process look like? You come into an IND, and then you can start what are called phase 1 trials. The process is phase 1, phase 2, phase 3, and then NDA, and then approval, and then launch. Phase 1, I'm going to give you some gross generalizations, but just to orient us. Phase 1 typically takes about a year to run, costs single-digit millions of dollars. Phase 2, 2 years to run, double-digit millions of dollars. Phase 3, 3 years to run, double-digit to triple-digit millions of dollars. So back of the envelope, just to do the clinical work through animals, humans, and get to a point where you have a package of data that you can give to the FDA for approval and their blessing, it's going to be well over hundreds of millions of dollars. The other aspect you have to think about is the manufacturing. This is the only industry in the world where you have to have redundant, backed up manufacturing before you even know you have a product that's ever going to be approved. Makes no sense at all. But the fact is that once your drug is approved, your innovation is ready to help a patient, no one wants to wait. We want to scale it up and get it out there as quickly as possible. That means you're investing, again, tens of millions of dollars into manufacturing you may never use. It's a very unusual concept. But everything about your process, the clinical work, the manufacturing, that's all regulated. So that means you have to have people who are experts in managing the regulatory process, managing quality control, good clinical process, good manufacturing process, good lab practice. All these things have to be not only done, but monitored and managed. That's why Insmed is 600 people today. It's one drug. And it's sold, in the US, Europe, and Japan, but it's only one drug. And that infrastructure is enormously expensive. Since I joined the company 9 years ago, and this is not unusual, we have raised $2 billion of capital. Now think about that. We can have another discussion another time about why drugs are so expensive. We don't have oak paneling in our offices. It's not where the money is going. It's going into the clinical trials. It's going into the regulatory process. It's going into the global manufacturing and distribution. It's going into having redundant inventory, all that stuff. And so it's just, when I said at the outset, I don't want to dissuade you, there's an enormous amount of work that has to go from the moment the idea is ready to go into animals for testing. And so it's really important at that outset that you've got your hands around an idea that can have a disruptive, positive impact on a patient. And so the easiest way to start with a great idea is to think, what patient is this going to help? How is it going to change their life? And does it pass what I call the holiday test? When you're done with the drug, someone in your family, God forbid, has the disease. In front of your family members, you slide it across the table and you say, this is going to make a difference. You can pass that standard in your own mind, then you know you've got something. And if it's just marginal, if it's the 10th statin, if it's an extra version of something else, it's not enough. My challenge would be to go further, go for something more disruptive. And this is how we segue back to Chris and Carl, because the work they're doing is truly disruptive. It's trying to get into and modify proteins for exceptional therapeutic benefit to patients in a huge variety of diseases. What that really allows us to do, they have created an engine, a platform from which we may produce a whole variety of impactful programs, right? So this technology is high risk. This technology is also very exciting because they've done some early work in validating it. So I wanted to bring the two of them in to talk a little bit about how they got from their idea to the place where they could prove it. Because I think that's probably the bridge that's the most interesting to the people that are assembled here. So maybe, I don't know, Chris and Carl, I'll just throw it over to you. When you were first thinking about this idea, how did you understand the journey from what is a brilliant academic concept, turning that into something that had that practical dimension? Carl, you mentioned about the practical side. Maybe you could address that first, and Chris, by all means, please chime in.
Yeah, so if I step out of line, Chris can insert himself and get us back on track. But you know, I think this is actually one of the things that I would encourage all of you to do is to, you know, go and talk to your colleagues who are either going through these types of entrepreneurial translational experiences or have been there, because there's a lot of lessons learned that I think can help you leapfrog some pitfalls that are hard to avoid without talking with people that have more experience. So, Will, to your point, I think that Chris and I, we bootstrapped our way a long ways, and I think that slowed us down. So, you know, we kind of had a kernel of an idea for one of the things that we now do, which is thinking about the problem of biotherapeutic immunogenicity. So if you take a protein and put it into a human being, our immune systems have evolved to surveil those types of foreign antigens. And there's a risk for any protein that a patient's immune system can mount a response against that protein. And there's all kinds of detrimental clinical consequences that can come from that. So this is one of the things that we have thought about for a long time. And when we first started talking, when I first got to Dartmouth, we kind of had a kernel of an idea about how we might be able to leverage Chris's computational expertise to tackle this problem more effectively and more efficiently and faster than it had previously been tackled. But I think that as we got started on that, we definitely were wearing academic hats, and we started doing what I think was really important foundational work. But we were working with some model systems that were fantastic for an academic career, kind of fantastic for, again, laying this groundwork and proving some fundamental concepts. But at the end of the day, I think that there, you know, was not really this impactful translational potential for some of our early successes. And so I think that's a really critical thing, maybe just to keep in mind. And certainly in the academic arena, you have to balance this, because sometimes model systems can be really productive, right? In getting papers and getting grants and getting success. But I think it's also really important to be thinking about what's beyond the paper, right? After you publish this paper, after you get this grant, what does the future outlook, what is that like for maybe this particular molecule or this system that we're working on? So it took us several years to kind of work our way through some model systems that were, again, very effective from an academic perspective, to finally thinking about and then tackling some target molecules where success maybe would drive a little bit more interest from the pharma world. Will, Chris, I don't know if you want to add anything to that.
Yeah, I think that's a really good summary. I think, you know, Carl put the nail on the head by saying, you know, we really approached it from an academic standpoint, and I'm even worse than that because I'm a computer scientist. So I saw a problem I wanted to solve, and that got me all excited just about solving that problem, without really thinking, okay, ultimately, how's this, as Will says, how's this going to impact the patient? And so we did have success writing papers and getting grants and all that. But I think we eventually got a kick in the pants when someone called me up, cold called, and said, hey, I read your papers. Have you ever thought about actually doing this with a person from biopharma? Have you thought about doing this for biopharma? And that was really, I think that was the first real wake-up call for me, at least. It's like, oh, this is not just a fun academic exercise. This could actually change treatment of patients. And so that really gave us the activation, gave us the energy to get across that activation barrier, and start moving into entrepreneurship. But I still think, as Carl said, that we bootstrapped it very much. We're like, okay, let's build up. And there are trade-offs there. And I think, to reemphasize Carl's point, talk to as many different people as you can and get as many viewpoints as you can, because we heard every conflicting idea about the best way to set up our company and what's the best strategy in terms of raising funding, versus partnering, being a service company, building a pipeline, all these kinds of things. And we continually weighed all these things to decide, well, what's the right path forward for us? And just like with research, that path changed several times along the way as to, what's really the right thing to be doing now. As we got more experience and a clearer picture as to how what we're doing could impact patients, we reevaluated at several points what's the best way forward. Back to you, Carl.
That maybe just to kind of round out that idea, in retrospect, I think that if we had to do it again, I would hope, I'll cross my fingers, that we might be kind of smarter and more efficient about this. And I think in particular, it really is what getting back to what Will said, which is like the problem, thinking about the problem. So Chris and I had identified this, one example of a big picture problem that needs a solution. And I think we came up with some good technology with how to address that problem and solve that problem. But kind of in implementing that technology, I think we could have been more visionary in choosing targets and molecules and systems to work on that may have accelerated our ability to bring the impact of that technology out into the real world. So it's been a long road for us, and I'm not complaining, because it's been a fantastic journey, and we're in a really good place. But definitely there are lessons to be learned along the way about how to do things most efficiently, and how to capitalize on the capital and the effort that you will inevitably be bringing in as part of these types of research programs.
One of the interesting things about the more innovative ideas and the more innovative research is the ability to prove them out becomes even more challenging, because not only do you have something that's innovative, but the very tools that would validate it are innovative. So the assay you may be using to prove that something has impact in a particular model may itself not be valid in the eyes of people who are using it as a tool. And that's actually an interesting challenge that plagues a lot of companies as they move products that are very promising forward. They say, okay, we've got this incredible impact on, and we're dealing with this right now. We have a small molecule that's targeting a DPP1, and the ambition there is to inactivate neutrophil serine proteases when they're released from the neutrophil when it's recruited. And the problem is that the neutrophil serine proteases and the way to measure them in both blood and sputum is all very novel. So when we say, we want to make sure that it's impacting the NSPs, how do we know that it is? How can we measure that? Well, here's the assay we've developed. Well, maybe that's valid, maybe it isn't. And the variability around that assay may influence whether we make the judgment to bring a program forward or not. We could interpret incorrectly that it's doing a great job or a terrible job. So there's pitfalls along the way, and I think this begins at the earliest stages when you're thinking of fundamentally what is the problem we're trying to solve and how do we prove that we're solving it. Let me take a pause there and just see, I want to make this as interactive as I can. If there are questions, please fire away.
So when you... let's have a microphone. Should we get a microphone?
You can really formulate your question in the time it takes to bring the mic over. And for folks...
So in your early academic years, how were those years funded? Was that philanthropic? What did you have a partner? What did that look like when you were in those early academic years?
standard grant funding mechanisms, starting with some fairly small NIH grants just for the basic computational and engineering, building up to NIH R01s. And then when we started our company Stealth on the Side, we continued kind of that path of SBIR, STTR to provide short-term funding to validate the basic principles and to start applying them to a range of different targets.
Carl, would you add anything to that? Looked like you were coming off mic. Okay.
All the old people ask questions, I guess. When you were talking about the importance of need, and right when you say if we're really going to apply this technology to a real world need, what's the role of the clinician? When did you first, John, or others start to get engaged with MDs who either said, "Yeah, this is insidious," or "Here's what doesn't work to date"?
Yeah. Well, what I would say in response to that is it varies by product and by idea. But in this particular case, once we had established enough support for us to add our own research to the work that was going into the singular focus of bringing our first drug forward, right? Once that was done, we could begin to multiplex. And that's the excitement that comes from a company that manages that process, is that now there are resources to go out and think about other things. And so we challenged the folks that had helped us create the molecule. Because you remember that journey I described is a decade or more in length. So once the idea is formulated, indicated it doesn't change, it has to remain the same, those folks that came up with the idea, I then very selfishly turn to them and I say, what's next, right? And so part of that is thinking what they can come up with, and part of that is going to conferences, having interactions at all kinds of venues, mostly academic or venues around like the American Thoracic Society or those kinds of settings, going and looking at the poster sessions, seeing what the new ideas are, reading the papers that are being written, and finding out what may be interesting. And there are people that then spend their time within our company just thinking about, can I bridge this idea to therapeutic benefit? And one of those people, a guy by the name of Sasha, actually went to a conference and bumped into Chris and Carl, and that was what began his curiosity in what they were doing. And he had an aha moment. If this works, this could be majorly disruptive across a whole range of areas that we were interested in. And that began that dialogue. Now, this is but one journey. Chris and Carl could have said, thank you for your interest. We weren't the only company around. We weren't the only source of capital. They could have gone to venture capital at that stage. They could have built their own company even larger than they already had. Lots of choices. Navigating that alone is tough. So one of the other pieces of advice I would give you is, as you're talking to that clinician, get somebody you trust, typically somebody who's done it before, who can help you navigate the choices of sourcing capital and thinking about company formation. Tech Transfer Office is a great place to start. But there are others, and I would include in that anyone you know that has built a company. A small company, a large company, find a mentor that you can turn to say, here are the options I'm being presented. Am I on the right track? So that they can get constantly guided back to what's the fastest path. I don't know, Chris or Carl, if you'd add anything to that, because you were on the other end of it. When did you start thinking about this with the mindset of wanting to talk to the clinicians that are going to be, thinking about this through the lens exclusively of the MD and the drug development pathway?
Yeah, so some advice that we got early on was you should be talking to clinicians early and often, right? Because again, you want to have a laser focus on the patient and what's the potential impact of this project that I'm working on. So I think that's good and solid advice. I think the flip side of that is it turns out that doctors are really busy, and when you have some harebrained idea, it can be like hard to get 5 minutes with physicians. But I think at least kind of getting a sense of which direction the weather vane is pointing from a clinician's perspective is super valuable early on. And I have to say that this is one of the really powerful things for us about now being part of the Insmed family, is that we have these types of physicians that, can really help think through what is the impact? How can the technology that we've got really help impact patients, and how do we think about translating things forward? So it can be tough early on as a small company, and I think I would just reiterate Will's advice that you've got to build a network of people and get introduced to people, because there are lots of folks out there that will volunteer their time and offer advice, but you definitely have to put some effort into finding kind of those mentors.
So we've got Michael Brookman, the CEO of Time Therapeutics on Zoom, is going to ask the next question, if that's okay.
Yeah, of course.
Go ahead, Michael.
Hi, thanks, Michael, for the opportunity and for organizing all of this. And next time I actually plan on being there in person. The weather was just a little too chilly to make the cross-campus trek. This is a really interesting story about Carl and Chris's translation of technology out of the university and raising SBIR funds and non-dilutive funding, and then eventually forming this partnership and being acquired. It's actually a path that we're heading down right now at Climate Therapeutics. We've been really successful at raising SBIRs and getting a lot of DoD funding. We're making fake platelets that help stop bleeding, so it's like a really easy thing for the DoD to want to fund. So I'm curious to hear your story and experiences and possible difficulties in transitioning from grant funding to investor funding, and how that transition progressed and challenges you encountered. And then how far along in the angel investor, VC investor pathway did you make it before beginning this interaction with Insmed and eventual acquisition? I can't recall exact details about when that happened and how far along the technology was, because and then if you want to comment about how your startup company grew with additional non-dilutive funding and eventual integration into Insmed. Sorry, I think I asked 5 questions, so this is a really exciting topic for me.
Chris or Carl, you want to take that on?
Yeah, so I'll kind of maybe take part of that, and Chris can chime in. And I think Will alluded to this earlier, that as well as Chris, that there are lots of ways to go about doing this, and I think you've got to find the path that's right for you, because there are benefits and liabilities associated with organically growing a company with SBIR funding and government funding, which is completely non-dilutive, right? And you and your co-founders own everything all along the way, which is fantastic. I think one of the downsides that really began to grate on us with that as we progressed kind of down the company's history was the pace at which you can advance when you are constrained by a grant that has a defined budget that you can rebudget 20% on, and sure, maybe you got a $3 million phase 2 SBIR, which is fantastic and super exciting when you get it, but then they're going to trickle that in over the course of 3 years, and time is everything when you're thinking about biomedical research. Time is super precious, and so I think those were things that we were weighing. We had been very successful as well at getting SBIR funding, and in fact, our success had really been amplifying right up to the point that we had engaged with Insmed. And I think that when we started having these conversations with Insmed, and we saw the fit with the company, which is maybe a separate topic, but we just saw that there was a good fit there. And then you think about the resources that a company like Insmed can bring to bear on these problems. It was just, I think, very clear to us that this was the opportunity, and this was the fit, and this was the time. And there has been no question in my mind along the way that this was it. And I think that's a really reassuring feeling. So I don't know if it's always that clear cut, because I've got an N of 1 at this point, but I think that you'll get a sense of what's right for you and what's right for your founders as you kind of go through the process. But it's good to be cognizant of the pros and cons of these different strategies.
Thanks. Yeah, we're running into this same, love-hate relationship with grants as far as, thanks for the money and thanks for the handcuffs as well. And it's great. We've been able to de-risk the program quite a bit through this process, and it's opened up a few introductions to potential partnerships that we're pursuing, actively.
Let me just make one comment if I can, because prior to these two companies that I've been a part of, I was the dreaded investment banker. And while I raised $2 billion at Insmed, I probably participated in raising close to 30 as an investment banker in a 10-year career. And that's not a lot of money in that world. The important point of that is my takeaway from that experience of all those companies, and it had to be more than 100, take the money wherever you can get it. I understand the attraction and the intuitive appeal of a grant, but if you can get more money sooner, I promise you this process is going to cost you far more than you would ever think in money, in time, in frustration. And the best answer for any biotechnology problem is having more capital. We have $926 million on the balance sheet at Insmed. And the reason we did that, we raised that money, was because we know there are massive problems coming. I have no idea what they are. They keep me up at night, honestly. Manufacturing disruption, suddenly a raw material is not available. We found out strange things like the wool grease from the sheep in New Zealand gives rise to the cholesterol that's used for the lipid that is used in our liposomal encapsulation of amikacin. Guess what? If they have a blight over there, we're suddenly unable to make drug. So buy inventory. That's a solution. Find another source. You got to qualify it. It's regulated. That's another solution. My point in this is whatever your problem, however advanced or early stage, capital is a great answer. And so it's difficult because you are surrendering control early, especially if it's a venture capitalist. No disrespect to venture capitalists, but I've worked with them. My first company was entirely venture funded. And I used to say we were successful despite their best efforts, which is a terrible thing to say, but it's true. There's strong opinions and they're very commercially oriented. You lose that feeling of connection to the patient that you can have when you are the master of your own capital and destiny. But that would be one thing I would add to what they're saying, which is get out of the cycle of incremental addition of capital. Don't worry about the dilution. If your program is successful and it has impact on patients, that's the real goal. If you get that right and you truly disrupt patient care for the better, you will be rewarded richly for that without even having to try. Yes, please.
I have a question.
Sorry, let's get the microphone quickly and then we'll get back to Batula.
I'm sorry, I didn't realize.
No, it's okay. You can introduce yourself as well. That'd be great.
I'm not used to people raising their hand. Yeah, don't do that.
All right. Hi, I'm Evelis. I'm a master's in regenerative medicine and entrepreneurship. I am interested in one day building my own biotechnology enterprise. And I would like to know, since we have the opportunity to have this panel of experts who've already done the work, if you come from scientific training, what would you recommend in order for you to create a biotech enterprise? Would you recommend somebody who only has a biology undergrad and who's right now doing a master's in biology and regenerative medicine / entrepreneurship? Would you suggest them to do an MBA as well, or is that not necessary? What kind of path do you think would lead to the most successful one in terms of preparation in order to get to where you are today?
So when you say in order to get to where you are, one of the things I want to go dig a little deeper on is what is the specific goal? Do you feel that your ambition is to get the program to completion or to construct a company? Because those are two different things. They can align, but they don't always. And so understanding whether you're, asking yourself, what do you really want to do? Like I did a JD and an MBA and I did some banking and all these. I'm a builder. I like to see groups of really capable people come together to solve really important problems. I'm never going to be the author of the idea. That's not my gift. And I'm not really interested in just doing finance. Doesn't interest me. But I'm happy to put people like Chris and Carl together with people who have lots of money for great ideas and convince them that they need to be together. And then I get out of the way. So what do you want to do?
I want to be a part of creating the idea, building that solution, and then marketing it and being able to build a company based on that solution.
And what that will be is the founder of the idea and the founder of the company, one and the same. And that happens a lot, especially in a knowledge-based enterprise like biotech. The challenge comes with scale. And so to get to that first accomplishment, my advice would be, particularly if you want to do both, find the best possible business mentor that can bridge the gap that you feel you have in some of those other areas. Sourcing capital, organizing a company, thinking about human resources. How do I create an org chart for what I'm going to build? What do I need? Those sorts of things you will get from somebody who has done it before. And if you envision yourself building a company of perhaps 50 people, find someone who runs a company of 50 people. If you want to build a company that's going to be much larger than that, start with the 50 people. Because if you can get that right, then you are given permission to go to the larger opportunity. You don't have to have an MBA. An MBA gives you connections, it gives you a different perspective, but it's not technical training the same way scientific training is in terms of what you'll be able to do with that. At least that would be my opinion.
Thank you.
I don't know if that's helpful. I don't know, Chris or Carl, what would you add? Because you went down the path of starting to build a company, and at some point you did so, and you brought it to a place, and then you said, okay, you know, we're gonna link up with Insmed as opposed to building an Insmed, which was an option for both of you.
I think, not to speak too much for Carl, but I think we both realized we don't actually have that training and background as much and kind of what it takes to really build up a company. And that's not our passion either, right? At least for myself, for sure. My passion is really solving the problem, having the ideas, figuring out how to get from here to there, and how can technologies help do that. And so running our own company for me was just kind of a stepping stone to further validate what we've done academically, to start pushing that towards something that would impact. And when we started, we didn't know which of these myriad different directions that would go, right? Would it lead to VCs coming in and helping us build up that company, at which point we would definitely want someone more skilled than we are running the company, right? And the VCs would certainly want that too, and install such a person. Or would it lead to, as it did, you know, us actually becoming part of an organization that already has all that in place, and so we can plug in and just hit go from that point. And we didn't know that, but I think, at least for myself, I knew that my goal was always to be the problem solver type of such an organization.
Yeah, if I just like added to that a little bit, I think it's important to recognize what your interests are, and Will, you were getting at this as well. Like, you need to find out what you're passionate about and what you're good at, and then the other thing that is really important in all of this is to understand that it doesn't matter how many degrees you have and how many years you spend preparing for this. You're not going to do a successful biopharma, biomedical company by yourself. And so I think you've got to recognize where your limitations are and be proactive about going out and getting people that have the expertise that you don't. So you don't have to fill all of the roles. You can't fill all of the roles. So I think it's good to figure out what you're passionate about and then find partners, collaborators, consultants, mentors that can help you fill the gaps.
Right. So thanks to the three of you for that. That's like equally parts like inspiring and also very practically useful. So thank you. I'm the CEO of Science First Partnerships. We are a software startup. We actually make software that helps academia connect to private funding sources, addressing a big unmet need. And I'm also, I really, something that really helped me was to find a business mentor early on and also just get into like real world sales experience. So that definitely helped kickstart the company. My question for you is, when you're looking at like early stage companies, when you're looking at people, maybe they have patents or maybe they're like me and they have a sales record. What are the big things you're looking for if you're interested in investing in a company?
Yeah, so I think the single most important thing is, can I draw a direct line between the idea and the impact on a patient? And then I'll start going through the cascade of additional analysis, which is, has anyone else traveled that path? In other words, are you coming up with a really interesting, disruptive idea that 40 other people who are very well funded are also trying to do? Because part of this is selecting the easiest path to have the greatest impact. And that's a compromise at times. There may be something you're truly passionate about changing. You look at it and you say there's lots of resource and lots of ideas that are going there. You know, you're taking on the competition as well as the opportunity to impact that outcome. Doesn't mean you shouldn't do it necessarily, but you should reflect on that. And I'll give you a good example. When I first came to Insmed, this drug device that they were developing at the time was focused on treating gram-negative infections that inhabit the lung. So this could be NTM, non-tuberculous mycobacteria, MAC or avium complex, abscessus. It was looking at Pseudomonas aeruginosa in cystic fibrosis patients, as well as non-CF bronchiectasis patients. It was looking at Burkholderia. It was looking at a lot of different opportunities. Now, I'll be the first to tell you, I didn't know a thing about pulmonary medicine. They may be the CEO, so, you know, VC judgment right there, right? It's a perfect example. Maybe they didn't really make the best choice. But the idea here is, how do you make that decision when you're looking at those opportunities? The first decision to be made is to focus. Pick one thing and get it done well. And as I looked at those different diseases, knowing nothing about them, I turned to the medical people and I said, of Burkholderia, of NTM, non-CF bronch, and cystic fibrosis, which one have approved drugs? All but two, right? So Burkholderia and NTM did not. And I said, okay, how many patients suffer from Burkholderia? Well, it's not that many. It's a handful. How many NTM? About 100,000 in the US. Okay, if I'm going to distribute $2 billion worth of investment and I'm going to distribute it across 1,000 patients, quick math tells you that's going to be an extraordinarily expensive drug. Would this drug warrant that level of investment? You have to look at it through the consequence analysis of what happens if it's not treated. And in that particular case, it would never justify the price. NTM had nothing approved to treat it. There are a cocktail of off-label antibiotics that are used. So I thought, what's the real problem? Show me the patient that's already taken all those drugs and is still failing and is still struggling. Now that's a harder hurdle to clear, but that was where I was able to line up and say, okay, we're not going after CF, we're not going after non-CF bronch, we're not going after Burkholderia, and we're not going after all of NTM. We're going to go after the refractory NTM patient population, the subset of patients that have tried everything else and have failed, and they're on their way to succumb from the disease. That's a problem we're solving. And everyone said, yeah, but it's really tough. Okay, then let's go for it. And that's how we decided it. It wasn't starting with the pathways of biology that we could have gone down. Those exist, and people have explored that. We have to take advantage of that understanding because it empowers the ultimate decision-making, which is, how can I have real impact on patients? So that's how I went about that process, is to think, draw that direct line between patients and the impact on the disease you're going to have. And that's exactly what we've done with Chris and Carl, and why we got so excited, because what they're talking about doing, de-immunizing proteins, just conceptually, if you think about the problems that occur in medicine and its use, treating people, and how the immune system, well preserved for thousands and thousands of years, becomes an impediment to improvement of healthcare. Can you work around that? They have some ideas that maybe they can. You know what? I'm all in. Because if they can do that, they can rewrite chapters of medicine. They can change the way we think about treating people with certain drugs that may already even be out there, that we can just redirect in a way that won't have the same side effect profile and negative consequence. And it may unlock doors to using treatments and vectors that could really have a profound impact on healthcare. And as I said to them, the first thing that matters when you're talking to a potential acquisition, fit. Can I trust them? Can they trust me? You got to cross that barrier first, because I mean, as I said to them, I know what we're getting. You're bringing your entire careers and all your capabilities and all your passion, and you're betting that we're going to sponsor that in a responsible way. That's a huge bet. They have asked for such trust, and I've got to get behind that. And so what I said to them is, look, this is breakthrough technology. We all know we only get one shot at it. So let's take 6. Literally. Let's multiplex what you're doing and try to across several pathways so that quietly we can see if we can make it work. And if it does, then we'll put our head above water and start dragging. But between now and then, let's get to the business of going down the path and seeing what we can do. But it's trust first and foremost, and impact on the patient in terms of thinking about the.
Thank you.
Yeah.
First of all, I really think this is fantastic because you brought 2 academicians to talk to us about how you move something from academia into really real product. And this is takes special interest because a lot of the time as an academician, you don't think of the important things such as IP and whatever. You know, academician, we like to talk, we like to publish and this sort of thing. So this is one of the things which our students should learn if you have an idea. So that's fantastic. I hope you learned a lot from that. You know, the other thing, what you are mentioning is about you need to look at what the product, what is there some competitor? Maybe just for the sake of the students, this is sort of the business plan. This is what they need to do. But one thing I don't know if with respect to the company, which I started a number of companies here in Cleveland. I want to ask you about raising money. Because in the East Coast or West Coast, a lot of people really, it looks like there is a lot of cash around and people can raise millions of dollars. Do you think being in the Midwest, since you've been here, is more challenging to raise money and this sort of thing, apart from the idea per se?
No question, but it's also your advantage, strangely. If you're in Cambridge, the money is everywhere. But part of the reason that flows is because the networks are everywhere. You're talking to other people who are already building companies. Basically, those mentors we were talking about earlier, those people who are experienced in having done it, they're all right there. And you can talk to them. There are natural places for that to happen, for you to meet people who've done it. But understand that here, your advantage is investors are lazy. And they're not, you know, I have many friends. My wife's an investor, so let's all understand I have nothing against investors. But they really don't want to get on a plane and come to Cleveland or to some other Midwestern place unless there's an idea that's really compelling. That gives you an opportunity because you're not being, from an investor's point of view, they want the novel idea. They don't want the popular idea. They want to get to the idea before the others get to it. And so if you have something, concentrate on how you're going to make your case that what you have matters. Think about gold standard models. If you're doing something in high cholesterol, you're going to want to use the New Zealand white rabbit model because that's what industry looks at to know whether or not what you're doing is going to have impact on a human patient. If you can get some data from that, you show up on the East Coast, you'll get their attention. In the meantime, you can get the work done here, and frankly, it's a lot more affordable. I don't really want to go over what it means to try and open and run a company in Boston right now. But you know, people will find the ideas that are worth finding for the most part. The tech transfer offices, the ability to network through others who have built companies, they're all going to come. Because when I want to go and find an idea case, where do I begin? Who do I call? I'm interested in something in pulmonology. I'm interested in something outside of pulmonology. Who do I call? Do I call the head of the pulmonology department? Do I call the Beale Institute? And I say, what's going on? And they'll say, you know what, we may not know. We're going to go find out. We'll come back to you with the latest and greatest. And that medium of exchange is really important. I come to Case because I'm on the board and because I grew up in Cleveland, and I know what's here. And I have been desperately trying to find something here that I can bring into the world that I live in, the industry. It's hard. I don't know if that answers it. Okay. Good thoughts or?
comments?
Yeah, and maybe if Chris or Carl want to make any final comments on Mahmood's question, and then we'll wrap up just because of timing.
I don't know that I have anything to add to that wisdom.
Well, maybe any closing comments, Carl or Chris, about your experience and the journey you went through, and anything you want to offer to folks who are perhaps in your same shoes, but a few years earlier in terms of where they are on the process, and advice you might give.
So I might just say that this is I can't imagine anything that you could do that's more fun or rewarding professionally than to think about how your science and research could translate, into the real world, right? And to make a difference if you're interested in drug development and biomedical devices, like helping patients. So this is a fantastic journey that you either are on or may embark on in the future. I think for the students, what I might advise, Will, is, you're there at Case to get your graduate, undergraduate degree, graduate degree, whatever it may be. And there's some boxes that you have to tick in doing that. But keep in mind that the stuff that you're doing there at Case, the projects that you're working on there at Case, maybe they're simply your degree, but maybe there's something more to that. And so be cognizant of that. Everything that you do, right? Be laser focused on the potential that you could have on impacting the world with what you're working on, even when you're in school. And part of that rolls into the advice that you've gotten here today, which is, don't go talk at conferences until you file for patent protection. So just be cognizant of the fact that it can be more than just an academic exercise in a paper, what you're working on now.
Yeah, and I guess I would just summarize almost the same point. What was kind of tossing around in my head, and it comes back to something Will said before. It is a change of mindset, right? Academicians are really good at thinking big ideas, tackling problems, but it's just it's not even a huge change of mindset. Just enough of a change of mindset to say, okay, where's that ultimately going beyond the paper, beyond grant funding, whatever? Where is that actually going? Where is it going to hit human health? And I think that was the thing that finally drove me from, and I could keep on going down that road of academia, but here's a chance to actually change my mindset just a little bit, working on very similar things. But now the ride gets, as Carl said, even more exciting, because now we're working on these things in a way that's going to take us somewhere that's really going to impact. And so keep on thinking big ideas, but keep that change of focus to say, okay, where's that ultimately taking, and vet them, right? And make sure those ideas are going to get you to a place that's going to have an impact in the translational world as well.
If I might just comment, actually, what's so exciting about this as an academician is one thing you mentioned before, which is, when somebody writes to you to tell you, thank you for this drug you have in the market and whatever, it's so exciting. And also when you are doing something which is really comes out from basic science, and then it really happens, you feel euphoria. It's something, a pleasure. But one thing you need to remember: this is not a walk in the park. This is a hard business. But if you are successful, you feel great. So that's, if I can tell you, a message to take with you.
All right. Well, thank you so much to the audience for these great questions, and a huge thank you to Chris and Carl and Will for this really insightful and applicable talk to everyone here today. Thanks so much.
Chris and Carl, thank you.